A very good morning, everyone, mostly our panelists and and family. Thank you for coming down in the morning. It's an early morning, and for everyone to be here is quite testimony for our interest in the area. Um, so uh, we made this session largely on innovations in Opthal, and all of our panelists will be talking entirely on different cutting-edge innovations that they're working on. Our first presentation is on CRISPR genomic editing, and just before we start off, I'm curious that how many of us are aware of CRISPR? Musaba, are you aware of CRISPR? Are they using it in Jordan yet? No. Okay. It's come out to be a very powerful DNA editing tool and holding a lot of potential. And if we look at therapies when it comes to retinal dystrophies or so on, we could perhaps broadly divide them into regenerative therapies or genomic editing, wherein you can edit the DNA, the mutation, and subsequently correct the copy of the gene, and the better protein is formed as a consequence of that. Versus you put in something which regenerates the dead or dying cells. So stem cell therapies largely take care of regenerative medicine, and this CRISPR genomic editing would take care of mostly whatever mutations are lying. So CRISPR largely has been a very powerful DNA editing tool, and it has a very high potential for ophthalmology and retina, to be specific, because we have a group of conditions, the retinal dystrophies, for which no treatment really exists. And CRISPR can potentially give us a cure for that. The advantage of the eye along with this is, of course, that it is a relatively immune privilege area and very accessible to intervention. For any other conditions, you would need to perhaps give it within the systemic supply or and it can be very hard to access particular organs, say for example renal cell carcinomas or something. And it's also very much possible to monitor any intervention. We can directly look at the eye, do the electrophysiology, measure vision, and so on and so forth. This has grown into be a very big multi-billion-dollar industry now, and the July of last year saw Allergan partner with Editas, and they're taking forward CRISPR genomic editing. So initially, while a lot of genomic editing was about using meganucleases or zinc finger nucleases, talons, all of these required significant amount of engineering, both of the protein, and it would take considerably longer period of time. What CRISPR has done has largely brought that down in terms of the size or the power of the enzyme which is used. And if I was to summarize, it largely consists of this. This is what the powerful tool is. The magenta thing that we're seeing in this diagram is the enzyme. The yellow strand is the RNA, and the blue, of course, is the DNA. So what this enzyme does is it scans the DNA, it picks up what area of mutation there is. The yellow strand, which is the RNA, which is connected with this enzyme, which is overall called the ribonucleocomplex. It binds with the DNA, and then once the match is there, like it identifies that this is the area of the DNA which is affected with the mutation, then it cuts the DNA. It initially started off as a bacterial defense mechanism, and basically bacteria would, whenever they would be infected by a virus, they would take in a part of the viral DNA and put it as a sequence within their own genome. And next time, whenever the virus would attack, they would use that same sequence and be able to destroy the viral DNA. So that was the roots of its discovery. And overall, a fairly shorter, a smaller protein RNA complex, it makes it very possible to edit the DNA using this. So while all of these are the advantages that it has, One of the biggest disadvantage is that it can edit mutations far away where it's not necessary as well. So these are called, off targets. So you don't want it to have an activity there, yet it might edit. And that can be a big disadvantage as well. You might induce mutations as a consequence of this. So to make it more predictable, it has been modified or fine-tuned even further. So the next tune of things has been the CRISPR base editors, and they largely, while you could think of the CRISPR, the initial enzyme to be scissors, the base editors are largely like a pencil and rubber model. They edit, change the one particular nucleotide, and then you can make the change in point mutations. So this would perhaps take it even more detailed. The top diagram kind of shows how all of this edit happens using a full double-stranded break of the DNA, whereas the base editors are just having a deamination domain. So they're able to deaminate adenine to a cytosine. I'll take some more time if that's good. So it's able to deaminate an adenine to guanine and a cytosine to thymidine. So that makes it very useful for any kind of transition mutations. So this is something which I perhaps had just dealt with right in the beginning, and this kind of enlists all the mutations that can be there. What makes overall use of this technology also very challenging, and perhaps an advent of precision medicine, is the fact that there is so many mutations that can be targeted. So my broad understanding previously would have been perhaps that a particular gene is involved and that gene is responsible for overall mutation. But then RP is very diverse, and there are 20, 25, or maybe more genes which can be involved, as well as different mutations in a particular gene. So RPGR, retinitis pigmentosa, GTPase regulator gene, is something which is responsible for X-linked retinitis pigmentosa. And it by itself can have all of these point mutations. The ORF15 zone is where you would have lots of insertions and deletions. So it just makes it very hard to perhaps make a cookie cutter technique for everyone. And that's where the advent of precision medicine, wherein you can actually make individual changes in the genome. Similarly, retinoschisis. RS1 is a full gene, but there are so many different mutations. So all of these potentially can be targeted using the CRISPR genomic editing. So perhaps the work is essentially to first sequence the patients, find out what mutations they have, then use their cell models to develop iPSCs out of it, and then use the CRISPR-Cas very precisely against their mutations. So our work is largely on cell models at the moment, and then eventually move into animal, and perhaps if that is showing promise, more towards the clinical side. So I think I would like to conclude on that note, just that CRISPR is a very, very powerful tool. Thank you. Go ahead, guys. All questions from you. Sagnik, you've been doing a lot of proteomics work, so I'm curious what you think of it.
Like, is it going to come as a intravitreal injection or a systemic therapy or a long-lasting implant or a one-time injection? How do you think it's going to translate?
Very largely likely to be an injection, so either intravitreal or subretinal. And systemic for sure not, because we don't want all the target to happen in all over the body. When we do look at germline mutations and that point of time want to change that, maybe then, but that's a very ethical issue. So for somatic mutations or very eye-specific ones, which we're looking at editing, there it will definitely be intraocular injection, most likely subretinal. Also, an initial thought for me was that, is it going to be at all useful? Will it change the mutation once actually happen, and how will it largely translate into delay in the condition, the RP that the patient's having? But to that, I strongly feel that it's likely to delay progression. Sometimes we have patients who come in at 612, 618, and all we tell them is that you can, there's nothing much that can be done, and you're going to have progressive blindness. If we are able to change the mutation at that point of time, and we are able to correct the proteins which are responsible, so it's likely that we will be able to prevent further progression, if not get the vision back. If someone who's at end stage, there it may not be of that much help, I feel. Oh yes, sickle cell anemia is a very strong use. The base editors I was talking about, so if you remember the valine to glutamic acid, single base change, so that's where it's gotten a very strong case. There have been a few clinical studies as well, and cancer therapeutics is going to be also another very strong area of use. Within Ophthal, the clinical trials have started for the first stage. This is for LC only, RP65 itself, I believe.
As compared to Luxturna, how much would be the cost reduction in CRISPR? Because as far as I know, it slashes the cost of gene therapy a lot. So any idea on the estimate of how much cost effectiveness it would have? Because that would eventually make it viable for widespread use.
Right. At the moment, I doubt that anyone really knows about the potential cost. Perhaps a relevant question at that note is the difference between gene therapy and the genomic editing. So gene therapy would be very, very useful for augmentation. It's a gene replacement or augmentation therapy, wherein you're putting a correct copy of the mutated DNA, and works very well for heterozygous conditions or homozygous recessive conditions, wherein both the copies are affected and you're putting one correct copy, and that's typically relevant for autosomal recessive conditions. But yeah, this will be most useful for X-linked or autosomal dominant heterozygous conditions as well. Great, relevant questions. Thank you. Let's have Sagnik now take on and tell us more about the proteomics aspect. Thanks.